was able to supply was that he had an isomer control. So just a single switch in the direction of the stereochemistry that renders the compound inactive against DYRK1A. And so what you can see is this isomer basically is completely inactive against Th17 differentiation. So giving us really, more confidence. And so still, let's see, that did not work. Let's get to here. Maybe that'll work. There we go. And so finally, we did a gold standard experiment. This took a long time because of technical details that I'm happy to discuss later. But what we generated was a conditional knockout for DYRK1A late in T cell development. If you delete it very early in development, you basically get no T cells that develop because it's very important for early development. And what we're doing here is really deleting DYRK1A after the T cells have developed. And so this is what I'm trying to set in mind, is that DYRK1A can have different roles in immature precursor cells as well as in mature cells, right? And so this was work led by two former technicians in my lab, Keegan Mu and Matt Maleg. And you can see that when you take a conditional knockout T cells, so T cells deficient in DYRK1A, they are less efficient in being able to generate these Th17 cells in vitro. We noticed some overlap. Matt wasn't terribly happy about that. And so we did an experiment where we said, okay, this is a 4-day culture period. Before we start any culture, we're going to take an aliquot and measure how much DYRK1A is in the cells, right? So we're trying to ask if this overlap is due to inefficient deletion, right? Because this is a pre-mediated deletion. And so what you can see, what Matt found was an eerily linear correlation between the amount of DYRK1A that the T cell expresses at baseline and the amount of Th17 that the cells are able to generate, right? And this overlap is really because there's overlap in the amount of DYRK1A being expressed, suggesting that DYRK1A is an analog, a tunable regulator of Th17 differentiation. So this suggests that maybe if you have more DYRK1A, you have more Th17, and that's exactly what we find here. We're using a mouse model that overexpresses DYRK1A only on a bacterial artificial chromosome. And you can see increased ability to generate Th17 in cells overexpressing DYRK1A. Now, we went and asked and took this one step further. We asked if this was due to cell intrinsic features. And so we generated a rag chimera, so basically immune deficient mice that we reconstitute with marrow, a mixture of marrow with both DYRK1A sufficient and deficient marrow. And so we can isolate the T cells from these mice, and each line is basically connecting the control versus the knockout T cells. We can discriminate between them using a congeneric marker. And you can find that in every single case, the knockout cells are able to generate less Th17 as compared to the control cells. So again, telling us that DYRK1A is regulating Th17 differentiation in a cell intrinsic manner. So we spent a long time trying to identify the mechanism and a long time of confusion. We could certainly see that if you look at canonical later markers of Th17 differentiation like ROR gamma T, this is decreased in the knockout. But if you look at early after differentiation, so this is only 2 hours after differentiation in a 4-day window, right? You can see that BATF, the regulator BATF, is decreased, but not the regulators MENA or POU2AF1. Again, telling us that DYRK1A seems to be regulating a part of the early Th17 program. We went through a lot of work to try and understand what was behind that part. We did a lot of RNA profiling studies with some limited yield, right? We could certainly, for example, generate a signature of what are the genes that are differentially regulated during Th17 differentiation. We could see differences by 5 hours. So what we did here was we said, okay, if there's no compound, what is the signature of Th17 genes? And we can identify the signature of about 4,100 genes, and we can ask which ones of these are dysregulated by harmine, and only a subset of them are dysregulated. So only about 482, and we focused on the 272 that are regulated in the opposite direction that we reason might be the most important. And if we do gene set enrichment analysis, one of the top pathways that comes out is some cytokine signaling pathways like IL-2 STAT5 signaling. Okay. Now, we finally figured out the biggest key when we did the experiment that we put off for the longest time, right? And so one of the key cytokine for Th17 differentiation is IL-6 signaling, okay? So IL-6 drives Th17 differentiation, and the way it works is it binds to the receptor. I'll give you more detail on this later. And this drives, in turn, phosphorylation of proteins, including STAT3 on the tyrosine. And this is really critical for driving Th17 differentiation. And we've not really paid a lot of attention to this for a long time because DYRK1A primarily phosphorylates serine residues, right? And so we couldn't think of why tyrosine, we didn't think that it would be phosphorylating the tyrosine. And so we really just put off this control experiment. But this really work by John Crispino a couple of years ago, and he showed that DYRK1A could phosphorylate a regulatory serine residue in STAT3. And this residue then, phosphorylation on the serine positively drives phosphorylation on the tyrosine, and this really gave us a mechanism to think about how DYRK1A might affect phosphorylation on STAT3. And when we did the experiment, sure, and lo and behold, what we found is if you look at cells from either knockout or controls, what you find is IL-6 efficiently induced phosphorylation of the tyrosine on both cells from both, but you see significantly less phosphorylation in the knockout compared to the control. Okay, so this is good. So there's actually defective phosphorylation on the tyrosine, and so we asked if this could be due to defective phosphorylation on the serine, and it turns out that the answer is yes. So when you knock out DYRK1A, you see less phosphorylation on the regulatory serine. So this is consistent with a story, you know, that this is with a kind of a story that DYRK1A regulates the regulatory serine, and this regulates the amount of phosphorylation you have in a serine. So after having put off this control experiment for several years, we went and did the other thing. We went to the other extreme and we did more control experiments, right? We know that IL-6 drives phosphorylation not only of STAT3, but also of STAT1 and STAT5. These are all important signaling molecules. And we asked if we saw decreased phosphorylation of the tyrosine residue on STAT1 and STAT5, and the answer is yes. Okay, and so we asked really, we asked if this was really due to a pattern, right? Is DYRK1A regulating phosphorylation of regulatory serine residues on all of these STAT proteins? And so I already showed you that this was the case for STAT3. And so what did we see in STAT1? So much to our surprise, what we found was absolutely no difference in regulation of the regulatory serine in STAT1. And in fact, there is no regulatory serine that's ever been described for STAT5, despite decades of work, right? And so this suggested to us that maybe this regulation by DYRK1A on the phosphoserine is not a generalizable paradigm, and we still couldn't account for altered phosphorylation on STAT1 and STAT5, right? And so this was particularly confusing because there's really not a lot of steps between IL-6 and phosphorylation of STATs, right? IL-6 binds to the IL-6 receptor, which then dimerizes and phosphorylates JAKs, which then phosphorylates STATs. And so there's not a lot to look at, and so we came up with, based on some loose historic data, that showed that the IL-6 receptor might be differentially localized in response to phosphorylation on serine residues. We came up with what many of my colleagues probably thought was a crazy hypothesis, that maybe DYRK1A is phosphorylating some components of the IL-6 receptor, and this is driving altered levels of the surface receptor, which then tunes the sensitivity to IL-6. Right, and so the IL-6 receptor is comprised of two different subunits. So shown here in orange is IL-6RA, shown here in green is GP130. And so we came up first with some data that showed that IL-6RA is modestly decreased in naive CD4 positive T cells. But much more significantly, what we could find was that this is GP130, the green subunit, is significantly decreased in all T cell subunits, subsets that we have ever looked at, not only CD4s, but also CD8s, right? So if you knock out DYRK1A, you get decreased expression of GP130 on the surface, right? This really leads to the opposite question. If we have more DYRK1A, do we see more GP130? And the answer is yes. Right? So suggesting that this is a mechanism by which DYRK1A is tunably regulating sensitivity to IL-6. Now, this is a busy slide, and the point is not to have you read all these different things. The point is there are a lot of arrows, and the point is IL-6. The point is we think that this is expanding the story of what DYRK1A is doing now way beyond Th17 cells, right? Because IL-6 alone is a pleiotropic cytokine, which means it does a lot of things. It's very important in many features, ranging from aging to autoimmunity, infection, inflammation, bone health, gut health, so on and so forth, right? So this is really asking, right, is this effect on IL-6 signaling an important axis in health in people with DYRK1A syndrome? Moreover, GP130 is a subunit of an entire family of cytokines. Yes, it's a subunit for the IL-6 receptor, but it's also a subunit for the receptors for many other different cytokines, including important ones like the alarmin IL-27, oncostatin M, LIF, many others, right? And so this really raises a paradigm by which DYRK1A might regulate response to many other cytokines. Okay, and so really in this part, I hope to have shown you that DYRK1A is an analog, a tunable regulator of Th17 differentiation. And this really leads us to be interested in trying to understand how DYRK1A deficiency might impact immune function, particularly in people with DYRK1A syndrome. And that's a project we'd really like to kick off, right? Clinically, does this manifest as autoimmunity, infection, and gut homeostasis? Which of these are affected and how? Right, and led by our finding that DYRK1A regulates expression of GP130, right? This really leads to a larger question of how DYRK1A deficiency impacts signaling of the entire IL-6 family cytokines, and how this has impact on the function of immune cells as well as other cells, because GP130 is very broadly expressed. So that's a project that we're really interested in getting into, and I want to give you I want to give you a flavor of how we're attacking these types of questions in terms of human immunology. By first, I'm going to share with you some more supporting data. So this is proteomics data that was shared by Anna Fultzer and Oliver Glass and Kellen Weston. And so they shared their SomaScan data, and I took a swing at it. And one of the simple, asked a very simple question, right? So I took statistically significant proteins that were statistically significantly altered with a threshold cutoff of fold change of greater than 1.5. And I just simply said, look, tell me about the pathways that are enriched here. And here what we find is the most significantly altered pathway is really cytokine signaling, followed by interleukin signaling, which is, I don't really know why this is different from cytokine signaling, but fine, right? Because interleukins are all cytokines. So again, supporting this idea that this kind of signaling through this GP130 family might be really important, right? And if you look at proteins, if you just manually curate proteins and you ask what cytokines can I recognize that are significantly altered, what you find, you can easily find a list of 4, and 3 of them are GP130 family cytokines. Again, suggesting that this axis might be important in people with DYRK1A syndrome. And so to give you some sense of how we're thinking we might attack this problem in people with DYRK1A syndrome, I'd like to share with you a story of another community that I serve, which is people with Down syndrome, right? Which is, as you heard of, in people with Down syndrome, this is the most common chromosomal disorder, which is a 3rd copy of chromosome 21. And you basically have a triploidy of DYRK1A along with, yes, a 250 plus other genes, right? So it's not simply a DYRK1A overexpression, but I think it serves as a lens. What I'm going to try and make is the argument that we can use it as a lens to better understand immune changes in people with DYRK1A syndrome, particularly when we're starting off with smaller cohorts. And I'm going to give you a sense of how we use our immune analyses as lenses to gain insight beyond in multiple settings. Now, people with Down syndrome have a lot of immune changes, and I want to highlight that I had gone through top 10 medical school and medical training, and I say that to point out that I had gotten zero information that immune dysregulation was actually important in Down syndrome. So again, trying to highlight that sometimes these things might be hard to recognize. It actually takes work to go in and recognize it and trying to understand. And so that's why I think there's also value to try and understanding if immune dysregulation is an important component to people with DYRK1A syndrome, right? And in people with Down syndrome, they have increased autoimmunity, increased susceptibility to infection, and impaired response to vaccine as simply 3 of the very clear immune-linked functions that are altered. Okay, this is data from Joaquin Espinosa's group. This is RNA profiling data, and this is really to say that DYRK1A is expressed in basically every cell type. Each color is a different cell type, and if you take people with cells from people with Down syndrome, in every cell type, expression is overexpressed of DYRK1A is overexpressed in people with Down syndrome, and in T cells in purple, DYRK1A is particularly highly expressed. So what else is consistent with DYRK1A being this tunable regulator? Is it also relevant in humans? So consistent with that notion, in people with Down syndrome, you have more DYRK1A, you also have increased phosphorylation of STAT3. You have more DYRK1A, you also have increased Th17 cells. So this is also supportive of this DYRK1A IL-6 axis being important in humans and being regulated in humans. Now, we've been able to take this much further than that, right? So one of the projects that we've completed is published a couple years ago, is trying to understand how the immune system changes, the architecture of the immune system changes with age. And we wanted to and what we did for this project is we put together a cohort of 28 individuals with Down syndrome over a wide range of ages, that's specifically selecting for individuals without autoimmunity. We have control participants and participants with type 1 diabetes. This is a representative autoimmune disease that is not treated with immune modulators, so I don't have to worry about drug effects, right? And we do mass cytometry and many other analyses. So this is basically looking at about 70 proteins a cell, about 1 million cells a person, and we build a brand new computational pipeline to analyze this. It's a unique pipeline that allows us to build the highest resolution atlas of the immune system of people with Down syndrome to date. And we can identify with full false discovery correction about 651 differences. And you can see the immune system of people with Down syndrome is very different from that of controls. Okay? And the point of this is to say, again, more evidence that IL-6 is important. Okay? So if you look at the studies of immune function in people with Down syndrome, 99% of that literature revolves around interferon, because 4 of the 6 interferon receptors are on chromosome 21. And so the question is immune dysregulation in Down syndrome all about interferons? Okay, so we can see many of these inflammatory cytokines are increased in people with Down syndrome, including IL-6. We'll get to that in 2 clicks. What we've been able to do is build a very easy to interpret map of exactly what markers are dysregulated in which cell types. Here we are looking at CD4 cells, from the most naive on the left to the most memory on the right. You're basically looking for where you see a lot of dark colors and a lot of stars, right? So dark color means the darker the color, the bigger the change. Red is up, blue is down, star is false discovery significant. And so what you can easily see is naive CD4 cells are very phenotypically dysregulated. And then we did a simple experiment, really in anticipation of reviewer requests. Does interferon drive any of these differences? So we take control PBMCs and we treat them with interferon and ask, can we reproduce any of this? And the answer to our surprise was no, right? And we said, oh, maybe this is a lousy experiment. And so at the same time, we had done an experiment just for us, right? One for the reviewer, one for us. We treated the cells with IL-6, and to our surprise, what we found was we could recapitulate 3 of these differences. So some, but not all. Okay, and the point of all of this is that IL-6 is an important regulator of immune function in people with Down syndrome, and I would expect potentially also in people with DYRK1A syndrome. right? And again, this is consistent also, this idea of seeing three, some but not all, right? Is again, immune dysregulation in Down syndrome is not all interferon, it's not all IL-6, it's complicated. But I'm gonna get to how we think we can use this work as a lens to better understand immune dysregulation in people with DYRK1A syndrome. With regards to autoimmunity, we also looked at the literature, curated a list of about 38 38 immune subsets altered in people with autoimmunity. Okay, then we asked a very simple question. What happens to these subsets in people with Down syndrome who don't have autoimmunity? So each of these dots is one of those subsets. What you can see immediately is every single subset is altered in people with Down syndrome. It's below the line of statistical significance. Some go up, some go down. What you find is every single subset is dysregulated in the same direction as people with autoimmunity. All the red is on the right, all the blue is on the left, okay? So this is really telling us that this third copy of chromosome 21 is sufficient to remodel the immune system into a shape that looks like that of someone with autoimmunity, even before they've developed autoimmunity, right? And so what is the role of DYRK1A in this? How does this affect immune architecture of people with DYRK1A syndrome? That's something we're very interested in. We're also learning that aging is important and altered in the immune system of people with Down syndrome. So one of the reasons we started down this whole venture was to build something that my friend told me was crazy and wouldn't work, and so that's why he did it, to prove me wrong, right? And so what I wanted to do was build a linear model to show how the shape of the immune system changes with age. And this, to my friend Gautam's point, this has historically never worked well, at least. But I reasoned that one of the reasons maybe it didn't work well is because if you come in with your 10 favorite subsets, because those are the 10 subsets you think about, and those 10 subsets don't change linearly with age, then you can never build a good model. And so the point was, well, I'm not smart enough to know what the subsets are, or maybe I can use a program to help me identify them. And so that's exactly why we did this. And so, we, using our unique tool, we can identify the subsets that correlate the best with age. And then we can, using this, not surprisingly, then we can build a linear model that describes very well how the immune system changes with age. Okay, so this is all trained on control data. Okay, and because this is a linear model, we can now immediately substitute in data from our participants with Down syndrome. When we do that, what we find is that the immune system of someone with Down syndrome, on average, looks like someone without Down syndrome who is 5 years older, so advanced immune aging. Okay, now we can play with this approach, so inspired by approaches like gene set and pathway enrichment, right? We can say, instead of starting with all 300,000 subsets, what do we start with the subsets that are different in people with Down syndrome versus controls? This is our Down syndrome lens model. It incorporates a flavor of Down syndrome biology. It's still trained entirely on controlled data. It still works very well. And this now shows advanced immune aging in Down syndrome of almost 17 years. The more interesting point is that it is this Down syndrome lens model that specifically reveals advanced immune aging in people with type 1 diabetes, but not our original control model, right? So this is suggesting that there's previously unrecognized pathobiology of Down syndrome and type 1 diabetes, and gives you some insight into how we're thinking about being able to use Down syndrome as a lens into which to better understand immune changes in people with DYRK1A syndrome. And so the idea is leveraging our experience and larger cohort of data from people with Down syndrome, we might be able to ask, can we plug in immune changes of people with DYRK1A syndrome? Initially, we'll have smaller cohorts, so that it'll be noisier, but the idea is to be able to say, can we look for changes, for example, that might be in the opposite direction of people with Down syndrome, and focus in on what unites those findings, right? What is the unifying theme behind those immune changes? And so I just want to leave with one last little thing, which is to say that we've pushed this, push the boundaries to look beyond the global immune system, to look at antigen-specific cells. And so this is looking at a vaccine response study. We look at people with and without Down syndrome, both before vaccination and early and late after a COVID vaccination. And we can look at not only the immune system in the periphery, also look specifically at antigen-specific B cells and antigen-specific T cells, and we can find unique changes in those that you cannot see in the global immune system. And so we think that these types of efforts are really important, and we're eager to apply that to understand more about immune response in people with DYRK1A syndrome. So people with Down syndrome exhibit DYRK1A overexpression, broad immune dysregulation. These include changes that you'd expect. Some of the increased IL-6 response, right? Increased STAT3 phosphorylation. So it all makes sense. It supports that DYRK1A is important in regulating immune function, and that we're probably going to find differences in people with DYRK1A syndrome. Right? And so this in turn supports the importance of better understanding how immune health is altered in people with DYRK1A syndrome. And I've given you a teaser of how we're thinking of using Down syndrome as a lens through which to quickly understand the key immune changes in people with DYRK1A syndrome. This work is first and foremost, I have to thank the participants and their families. This is us at an outreach event. A tenet of our lab is we do this work in partnership with the community. We pay back to the community, including webinars, both of not only anything that we've learned with samples that they've donated, but also in helping them parse scientific issues that we may be of assistance with. I'd like to thank my funding, and I'd like to thank you for your attention. I'm happy to take any questions.
Sorry, going back to this GP130 contractor, I'm very impressed by this, the fact that it's being down-regulated in the syndrome. Have you checked the other pathway, other cytokines, and in this syndrome, down syndrome, is it overactivated?
Yeah. So I'd love for you to be on my review committee. Right, we have a grant in to do exactly that. We're trying to look for GP130 expression in cells from people with Down syndrome. The tools are terrible. So the flow cytometry antibody sucks. And so we're trying to solve that problem right now. We're also trying to work on exactly, like you said, like the entire, to ask exactly response to all these different cytokines, right? What's important, what's not. We know that this is really important because SNPs, there are polymorphisms in GP130 that are linked to diseases, many diseases, including autoimmune diseases. And so we hope to use this as a broad way to think about not only, I think we talk in very general terms right now, right? Like all people with Down syndrome, all people with DYRK1A syndrome, and I think that's where we start. But ultimately, also to understand the inter-individual heterogeneity, right? So how do all these things, core pathways, interact with individual genetics? It's a great question.
Sorry if I missed that. Heterozygous, their immune effects of these.
It is total. We've not looked at the hemizygous yet. So that'll be my dinner conversation with Jan.
So in your work, correlate that with like the same, as you mentioned. And so we have that many
Yeah, we're starting again. Would love to have you serve on the grant review committee. The short answer is yes. The short answer is yes in progress, right? I think what you're talking about is exactly the most important type of question to answer, and it's also the one that needs the most work and the most scope. right? As you know, like, because it's part of that, it's a question of, so what we're trying to get at is, I can give you an answer in two parts, okay? The first is we're actively working on that, and we're close to getting the answer with something limited like COVID vaccination, right? Because that's something that you capture a person at a known time point, you have a lot of control, right? It's not just, I think they had the cold 3 times last year, you know what I mean? That's a lot more challenging. But also we have ongoing kind of immune health projects that are really setting the pipeline to be able to do kind of what you're saying. You know, you follow people daily for 3 years, and you can ask after that, what are the immune sequelae? Thanks.
You. While we're getting set up for Jeremy, I'll just put a plug in. So I think this will be the last talk, and then I'll do a quick flow.
Okay.
And
Which one's yours again?
this one.
Do you have a pointer?
Yes.
It's right here. It's this one.
Well,
Yes.
that's
Good.
put a plug in there.
And it,
The other way.
should I turn it on? So,
Is it working?
yeah, okay.
Okay, well.
Okay.